Hi guys. So today we're going to talk about the immune system and I'm not even going to play. Today's video is going to be a doozy. So I strongly recommend you take this in chunks. I would not try to do the whole video at once. It's going to be a booty kicker. There's a lot happening in your immune system. The immune system is amazing and it's super cool, but it's also super complicated. It's so complicated that this is going to seem crazy, and this is only the surface of what your immune system can do. So I'm going to super duper simplify what's going on in here. Just know that it's way more complicated than what I'm telling you. Okay, so for starters, what is our immune system? So our immune system is the way that the body defends itself against foreign organisms, so whole living things that come into our body that we don't want in our body, and then also molecules on the surfaces of those organisms. And those molecules on the surfaces are known as antigens. So we might be talking about bacteria that come into our body, we might be talking about viruses, we might be talking about other cells. And each of them has little proteins on the outside of them that help us identify them. Those little proteins on the outside are considered antigens. Now, if those things that come into our body cause a disease, they get a special name. So all kinds of stuff comes into your body and doesn't cause a disease. Okay, those are just antigens. If they cause a disease, then we call them pathogens. So an antigen that causes a disease is a pathogen. So for example, a virus that affects humans is a pathogen for humans. But a virus that affects birds is not a pathogen for humans. It is a pathogen for birds. So unless it creates a pathology, so an illness, it's not considered a pathogen. Okay. So some basics about how diseases spread. You already know that when we cough and we sneeze, we leave tiny little droplets, minuscule ones you can't even see. Tiny little droplets are left behind, and the bacteria and the viruses, they can get inhaled by other people. They can get left on surfaces, and so you go grab a doorknob that somebody else grabbed, or you go use, I'm going to make you all paranoid now, you use the top of the Purell that somebody else used the top of the Purell before they Purelled their hands, and so they left bacteria there. All kinds of stuff. Diseases spread through that. Physical contact, body-to-body contact, so skin infections are an example of that. An exchange of bodily fluids, so blood or sexual contact, contaminated water or food. Things like food poisoning are going to come from contaminated food. And then there are infected animals, and an infected animal is called a vector. So West Nile virus is an example of one. The mosquito would be the vector. So the West Nile virus goes into the mosquito bites you, you get the West Nile virus. Rabies is an example where the raccoon or the whatever the rabid animal is would be the vector that's going to get the virus into you, or the bacteria into you, whatever the pathogen is, into you via an animal of some sort. Okay, so the first thing is that we're going to sort of divvy this up into two parts. We have innate immunity, which just comes from within, and it's a non-specific defense. There are internal innate immunities and external innate immunities. So we have non-specific defenses that are inside of us and outside of us. Then we also have acquired immunity. And acquired immunity is, as it sounds, acquired. We get it throughout our life. We're not born with it. That's going to be a really specific response. This then gets divided into two parts, the humoral response and the cell-mediated response. So if you can imagine a graphic organizer, you have immunity, you have innate immunity, you have acquired immunity. Within innate immunity, you have internal and external. Within acquired immunity, you have a humoral response and a cell-mediated response. I got bad news for you. We're going to break that down even further. So you will have a doozy of a graphic organizer by the time you're done. Okay, so innate immunity doesn't care what the invader is. It's a non-specific defense. So the external defenses, their whole job is just keep everybody out. Like, we don't even want to deal with you. If you can't come in, then we don't have to deal with you. So we're just going to keep you all out. Okay. So your skin is one of your external defenses. It's a physical barrier. It just physically prevents things from getting in. But then you also have acidic secretions that are natural antibiotics in your sweat and in your tears. So those things are actually going to work to kill off different microbes that are trying to get into your body. You also have mucous membranes. So in your throat, inside of your mouth, in your nose, you've got mucous membranes in all of these places. The mucus is really thick and gooey, and that's going to trap invaders. So they're just going to get stuck in the ooze. And then there's also lysozyme, which is an enzyme. That's an enzyme. It doesn't end in ase this time. It ends in zyme, but that's handy. So lysozyme is an enzyme that's going to kill bacteria. You have cilia in your respiratory airways. So in your throat, you actually have little hairs that are going, get out, get out, get out. And they're trying to wave all of the gross stuff out to keep it from coming in. You have stomach acid. So stomach acid can actually kill off a lot of the bacteria that come into your body, and digestive enzymes will digest those bad boys. You also have a fever. So this is internal now. So your fever is going to keep bacteria from reproducing because the temperatures are too high. And it's going to increase what we call phagocytic cell activity. Now, you don't know what that is yet. I'm going to tell you what phagocytic cell activity is, but I'm going to tell it to you in a little bit. So just a few minutes and you'll know what that is. But fever makes that happen faster. Okay, so when we get there, know that when it's hot inside your body, it's going to make that activity happen faster. So the fever is going to help keep the bacteria from reproducing, but then it's also going to help the rest of your immune system work a little bit faster. Okay. Okay, so also inside, this is a thing that creeps me out. It really weirds me out. So it's cool for me. Like, I have no problem thinking about blood vessels and that, like, you have this whole network of blood vessels going through your body, of your veins and your arteries and your capillaries. But side by side with all of those, you have all of these lymph vessels. So it's like a second whole set of little tubes throughout your body that are carrying this stuff called lymph, which is a fluid that is useful for your immune system. So you've got all of these different capillaries, and then you've got the lymphatic vessels. And so you can see in this diagram here, you can't see because I can't touch because I don't have a working stylus, but you can see in that upper right-hand corner of the diagram, there's the lymphatic vessel that's green, and then you have the capillaries that have the blood in them that's red, and everything is all just in between. And then in various places throughout your body, you have lymph nodes, which are places sort of like gathering points, junctures, and then you have vessels coming out from there. So your lymphatic system is another part of your innate, so non-specific, you're born with it, internal immune system. So what happens is that lymph is going to circulate through the vessels. It's going to go through the nodes. It's going to go past all the various organs like your spleen and your adenoids and your tonsils and all of these other things. And it's going to collect the microbes and the toxins. So it's just going to scoot by and it's going to grab all that stuff that's not supposed to be there. Kind of like a little garbage man, but it's like a garbage man fluid. It's like your sewer, basically. So your sewer is going to grab all that stuff, take it where it needs to go. There are also cells in there that are going to help you with both your innate and your acquired response. So they're also going to deliver guys who are going to fight off all the bad guys that are in your body. So your lymph is really cool because your lymph is this slushy liquid. Okay, it's not slushy. It's this liquid that's going through and it's picking up all the gross stuff, but also delivering fighters to where they need to get to. And it's separate. Your lymphatic system is separate from your circulatory system. It just sort of intertwines in between and everybody kind of shares and diffuses back and forth. Okay, so phagocytes. Remember I said increased phagocytic activity? Here we go. Phagocytes. So phagocytes are white blood cells. That's what WBC stands for, is white blood cells. And their job is to engulf and destroy invaders. So white blood cells are just going to come up and they're going to go and they're going to eat whatever invader is there. They don't care. They're like, you don't belong here? I'm eating you. Done. There are two kinds of phagocytes. There are neutrophils and macrophages. Neutrophils are in your bloodstream. Macrophages are in your lymph nodes. They're in your interstitial fluid. They're in your tissues. So they're in between. They're in lymph. They're a lot bigger. So neutrophils little, macrophages big. Okay. So in this category of phagocytes, you have neutrophils and macrophages. They are both eaters. They both have, they've got phage. Neutrophil doesn't have phage in it, but macrophage, big eater. Okay. Phagocyte, eating cell. Okay. So you can remember what those words mean. So phagocytes could be little, they're neutrophils, or phagocytes could be big, macrophages. If they're in the blood, they're neutrophils. If they're in the interstitial fluid and tissues, they're macrophages. Okay, so here's an example of this blue dude is our macrophage, and he's going to come along and go gobble, 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 and eat up those viruses. Okay. All right, these are cool. I just like their name. They're natural killer cells. I just love like the thought of what those would look like. So natural killer cells So what happens is when a cell is starting to get sick or when it's starting to get damaged or worn out, it releases different chemicals, and it tells that these other cells that they're stressed out. Okay, so they release these stress chemicals, and natural killer cells come by and they're like, oh my God, you're stressed out, and then they kill them. Thank goodness we don't have those for humans. Goodness gracious. And before things can get out of control, before anything really bad can happen, natural killer cells can come along and just go, oh, you know what? Sorry, collateral damage. You're gone. And so natural killer cells will come in, recognize a stressed out cell. They don't need to know it's stressed out. They don't care. No, they're like, you're stressed out. You're out of here. We don't have any time for stressed out cells in here. We're going to get rid of you. Natural killer cells will do that. So again, this is why they're non-specific. They don't care if it's a bacteria or a virus or a fungus or what it is that's bothering you if you're that cell. If you're a stressed out cell, natural killer cell, taking you out. All right, there are also these proteins called interferons. And you know what they do? They interfere. So they're proteins that get in the way of viral reproduction. So interferons are specific fight against viruses. But they're still really general. It doesn't matter if it's a cold virus, if it's a flu virus, if it's a herpes virus, if it's any kind of virus. They don't care what it is. But they do get in the way of viral reproduction. So what happens is the infected cell sends out the interferon signal to its neighbors. And then the neighbors now know, oh my God, wait, I'm going to make this new protein that's going to keep the viral RNA from replicating. And then that way, maybe that one guy sent out the signal, that one guy died, but now all the neighbors are okay. And the neighbors are already able to stop the spread of infection because they got word, because that first cell sent out the interferons, got the word to them saying, hey, make the protein so that you don't get sick from this virus. And then there's the complement system. In the complement system, there are proteins that cause a chain reaction. And that chain reaction is going to result in invaders getting killed. Okay, so complement system consists of a bunch of proteins. Okay, that's all you need to know. Bunch of proteins. They can do a bunch of different things. They can enhance phagocytosis, so they can make it easier, faster, better, stronger for the phagocytes to come chomp up invaders. They can recruit more immune cells, so they can get other immune cells over. So they're like, hey, over here, over here, over here, over here, over here. They can also trigger the inflammatory response, which we're going to talk about in a minute. So they can make it so that the inflammatory response happens. So the complement system is this whole set of characteristics that are going to complement all the rest of our immune system. So they're not going to be a standalone thing, but they're going to make it better. That's what it means when something complements something else. Not that they say, oh, you're lovely, but they are a complement. They balance it out. They help make it better. Okay, so the complement system, proteins, they're going to help all of these other things happen better. All right, so what is the inflammatory response? I'm sure you know about things that are inflamed. People have talked about inflamed. In fact, anytime you see itis in an answer, like otitis or appendicitis, it means inflammation of the something. So whatever it is that's got itis after it, otitis is an inflammation in your ear. Appendicitis is an inflammation in your appendix. So anytime you've got itis there, that's what that means. So damaged cells signal the release of this chemical called histamine. So histamine gets released, and histamine results in what we call vasodilation. So vaso refers to your veins. Dilation means that they're getting bigger. Like when you go get your eyes dilated, it makes your pupils bigger. So vasodilation means that the veins actually get bigger, okay? When the veins get bigger, the tubes get bigger. That's kind of like taking a garden hose and turning it into a fire hose. And so now instead of this much blood being able to get to the area, this much blood is able to get to the area. So you get this increased response of blood getting to the area. That means then that clotting can start. Clotting will get started faster because there's clotting factors in your blood. Phagocytes are going to get delivered. And so they're going to be able to come and they're going to be like, oh my God, where are the invaders? And go nom, nom, nom. And you might see some redness because of all of that extra blood flow. You might see some swelling because of all of that extra blood flow and that lymph there. So if you look in this diagram in the first slide, you can see that the histamine gets released. So this person has gotten pricked by this, needle or whatever it is. Those little orange guys are the pathogens and they're there. and little chemical signals go out from the cells. Oh my God, help, help, help. Notice in 2, you can see that blood vessel got bigger. So that blood vessel got bigger, the blood and the white blood cells can get there faster. And so they're going to go out. Clotting factors are going to go out. So that now blood is going to start to clot in that hole that was left behind by that prick. And so that clotting is going to go in, and that clotting is going to fill up that space. So now you don't have an open wound anymore. The phagocytes are going to go out, and they're going to start to gobble up, and they're going to munch all those little orange guys, whatever those bacterial or whatever the pathogen was that came in. And those phagocytes are going to eat them all, and now they're gone, and they're going to consume whatever other cell debris there is. So whatever cells died because of the pathogen or in the process of this. And then you can see that the cells are replicating and healing up the inside, filling that gap. And now you've got a nice healed wound. So the inflammatory response made it so that all of that could happen. All right, now settle in, because we have adaptive immunity coming on. So maybe you want to take a minute and go back and make sure you understand the innate immunity first before we go on to adaptive immunity. You might want to take a deep breath and then hit play. All right, so adaptive immunity is also called acquired immunity. It's also called specific immunity. There's a really good reason they have those names. It's an acquired immunity. You are not born with it. You get it throughout your lifetime. It's also called a specific immunity because what the pathogen is matters. So it very much matters if we're talking about a cold virus or a flu virus or a herpes virus or a botulinum bacterium or a clostridium bacterium or what it is we're talking about. So the very specific organism matters. And it even matters not just that it's the cold virus, but which cold virus it is. Out of all of the hundreds of cold viruses, your body has to know exactly which one it is. So this is a way more specific defense than, hey, you don't belong here. And that's what your innate immunity was. Your innate immunity was just, you don't belong here, we need to do something about you. Adaptive immunity is, oh no, we know exactly who you are and we got to figure out how to get rid of you. Okay, so you have these cells called lymphocytes. Lymphocytes, think about it, lymphocytes. They are created from stem cells in your bone marrow or in your thymus, and your thymus is a gland that's up here in your neck. So there are cells in your bone marrow and your thymus creating lymphocytes. Lymphocytes come in 2 kinds. There are T cells and B cells, okay? Well, 2 kinds that we're going to talk about. There are T cells and B cells. And then we're going to talk about different kinds of T cells and B cells, okay? But a specific antigen comes in, okay? A specific antigen comes in, could be a protein, could be a really big polysaccharide on the surfaces of the viruses or the foreign cells, and that's going to trigger the creation of the lymphocytes, okay? So a very specific antigen comes in, and that's going to trigger the creation of these lymphocytes, these T cells and B cells, okay? What you're going to end up with is the production of an antibody. Guess what an antibody is? It's a protein. It's a protein that recognizes and targets the antigen. So I like to think of antibodies as being little bullets. And so we shoot these little bullets out at these antigens to help get rid of these antigens. That's a super duper oversimplification, but that's how I like to think of antibodies. Just these little protein bullets that we shoot out toward these invaders that don't belong. You have to have a very specific bullet for every specific antigen that comes in. Okay. You have millions of kinds of B cells and T cells. Each of them has different antigen receptors, and each one of them is capable of binding with one specific type of antigen. Okay. So that means, so we said there's hundreds of cold viruses. So let's say there's cold virus number 1. You have a B cell and a T cell in your body that will recognize cold virus number 1. And they have the exact right protein shape, they have the exact right binding site to attack cold virus number 1, not cold virus number 2, 3, 4, 5, 6, 722, just cold virus number 1. Then you have a different set of antibodies and B cells and T cells in your body that can trigger antibodies for cold virus number 2. Now, those guys can't do anything about cold virus number 1. They can only work on cold virus number 2. And they can't work on herpes and they can't work on the bird flu, okay? So these guys hang out in your lymphatic system waiting until cold virus number one walks in. Cold virus number one gets in and they're like, aha, we know you. And then they go and attack. Okay. But they're very specific. So you have millions of them because you have to have something for every antigen that could possibly come into your body. Okay. So here's how it works for the first infection. First infection is complicated. Second infection, way easier. So we'll get through the first infection and then second infection, super easy. Okay. So the macrophage is going to come in. Remember, macrophages are going to come in and they're going to gobble them up. Macro, big phage eater. Macrophage is going to come in. Gobble, 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 gobble. They're going to digest that thing. And then here's the weird thing. They're actually going to display the antigen on their surfaces. So it's almost like if you go hunting and you wear the animal pelt. And so you're wearing now this thing that you've preyed upon. So this is called a self-non-self complex, which makes sense because, hey, I'm myself, but I'm also not myself. I've got this other thing on. And so the macrophage now, you can see that in the image, you've got your blobby sort of yellowish macrophage. It's going to eat up that orangey microbe, break that orangey microbe down into parts, and then display the parts on its outside. Okay, so now we call this an antigen presenting cell because it's a cell that's presenting antigens. It's going, hello, I've got an antigen on there. Would you like to see my antigen? So it's an antigen presenting cell. I present to you my antigen. Okay, so the macrophage comes in, gobble, 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 digest, 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 show the parts. Okay, we're good so far? I hope so. Okay, so now that macrophage is getting carried around and it's going to get carried to a lymph node. And when that macrophage gets to a lymph node, it's going to see these guys that are helper T cells and it's going to go, you guys, look what I got, look what I got, look what I got. I totally picked up this bad guy and I'm wearing him. And the helper T cells are going to go, oh my God, that's an invader and we've never seen that one before. We have to do something about it. Okay, so the helper T cell is the helper and it's going to be like, we've got to do something. And so what they're going to do is they're going to secrete these things called interleukins. And those interleukins are going to stimulate two types of cells, B cells and cytotoxic T cells. We're going to talk about both of those specifically. Okay. So macrophage, gobble, gobble, gobble. Oh, look at me. Goes to the lymph node. You guys, look what I've got. Helper T cells. Oh my God, we have to do something. B cells, we need you. Cytotoxic T cells, we need you. And they do that via interleukins. Okay. By the way, this is totally what's going on in your body right now. You just can't hear it. All right. So one more time, you can see this little green dude is a phagocyte. It's going to eat the bacteria. Gobble, gobble, gobble. Parts of the bacteria or the antigen go to the surface of the phagocyte. The helper T cell is going to see it. It's going to recognize it, grab on and go, oh my God, we need help. And the helper T cell is going to go, help me, help me, help me. And then it's going to alert the people, well, the cells that it needs to alert. Okay. Now, B cells. There are these cells floating around that are called naive B cells. They're also called parent B cells, but they're naive. They're innocent. They don't know anything. They haven't seen anything. They're so naive. And they're activated. Okay. And they're going to try to bind to the antigen. So they're going to go to where that antigen is, and they're going to try to bind to it. Every naive B cell has its own surface receptor protein sticking out of the membrane. These are called antibodies. Those little bullets I was talking about. Those are proteins, and there are genes that code for parts of the antibodies. So B cells contain different combinations of those parts. And so you can get different antibodies. So think about it this way. So think about you've got gene A, gene B, gene C. Okay. And I'm going to keep it as ABC to keep it really, really simple. But you've got millions of these things. Okay. Well, not millions of genes, but you've got different combinations that you can have. So let's say you've got gene A, gene B, gene C. Okay. So gene A makes this, gene B makes this, gene C makes this. So you've got A, B, C as your little chunks of proteins you can make. So you could have a naive B cell that only has A on the outside. You could have a naive B cell that only has B on the outside. You could have a naive B cell that only has C on the outside. You could have a naive B cell that has AB on the outside. You could have a naive B cell that has BC on the outside. You could have one that has AC. You could have one that has ABC on it. And then you can go out into all the rest of the letters of the alphabet. So you could have ABCD. You could have BC, BD. You could have DA. You could have ABCDE. You could have any, so you could have any combination, as many of these genes as there are, You can make these different combinations. One of these combinations is hopefully going to is going to make the puzzle piece that fits with that antigen. And so your body is just going to try all the naive cells it can. And so it'll work through every naive cell it can possibly create until it gets to one that works. This is why sometimes it can take a while to get well, because your body's cycling through all kinds of naive B cells and it can't get to one that's going to fit. Like, who knew that AQZP was the one that was going to work? But once your body finally got to AQZP, that was the one that worked, and it made the right shape to bond with that antigen. Okay, so here's a simpler version. So you've got these antibodies that are blue. Okay, you've got the marker molecules that are, on the outside edges that are the orange, and then the antigen itself they have marked as yellow. Well, anyway. So you can see that first antigen is going to work with that first antibody because that little circle is going to fit into the little circle space in the antibody. Okay, that second antigen won't work with the first antibody, but it will work with the second antibody. That third antibody is going to work with the third antigen. And then if you look at that fourth antigen, there are actually two different antibodies that could work with that one. So take a look and make sure you see what I'm saying when I say there are two antibodies that could work there. Okay, so the first one and the third one could both work there. The second one's not going to do anything for you. Okay, so you've got all of these naive parent B cells that your body is just going to keep trying, and say one of these has to work. Okay, a healthy adult can produce 10 billion different antibodies. 10 billion antibodies. So if an antigen comes into your body and you work on it long enough, you're probably going to find an antibody that will work for that antigen, because 10 billion, that was a lot. Okay, so here's another example, another way of looking at it. So you have the antigen binding site up there on the top, and then you have that main part that's the same in the middle. So antibodies are Y-shaped, and they just have that edge, that antigen binding site on those ends of the Y. Okay, so the way antibodies work is they attach to the invaders and they mark them. They mark them to be eliminated. So one of the things they do is they block the invader so they can't infect a host cell. So with a virus, they could grab onto the virus, and then the virus is not able to enter into the host cell. They could also cause the invaders to clump up. So they just make a big glob and then they can't go anywhere because they're in a big glob. And they can also activate the complement proteins, so they can actually make it so that those complement proteins are a little bit more active and a little bit more able to do what they need to do. Okay, so antibodies are great. They mark things to get eaten. I have that. Nope. Okay, sorry. So they'll mark things to be eaten. So you can see down at the bottom of this picture, you can see the macrophage. The macrophage wasn't sure whether it should eat that thing or not, but once it saw the antibodies, the macrophage was like, oh yeah, I should totally eat that, because anything with antibodies attached, that's an invader that needs to get gobbled up. Okay, I like this picture of this cell lysis, this rupture of this invading cell. I don't know, it looks cool seeing it explode like that, but that's awesome. All right, so remember what we had in our adaptive immunity. The macrophage went gobble, gobble, gobble. It ate up the dude, became an antigen presenting cell because it put those antigens on the outside of itself. And I was like, look at me. It circulated back, went to the lymph nodes, and it was like, look what I did, guys. And the helper T cells went, oh my God, oh my God, oh my God, we need help. And the helper T cells activated the B cells, and the B cells were like, we have to figure out what's going to work. And so the B cells started trying to find the right antibodies. And so naive B cell after naive B cell after naive B cell went, and they were like, I can do it. No, you can't. I can do it. No, you can't. I can do it. No, you can't. However many times, up to 10 billion, until they found the one that worked. And then they went, huzzah, we found one that worked. Okay. As soon as the antigen binds to a naive B cell, that B cell gets triggered and differentiates. And it's going to differentiate into two different types of B cells. Plasma B cells, which are sometimes called effector B cells, but I call them plasma B cells. Plasma B cells, and those plasma B cells secrete free-floating antibodies, those are the bullets I was talking about, that are going to combat the current infection. So the first B cells, the naive B cells, just had the little proteins out here, and they're like, hello, will this work? Okay, once they found out, they're like, pachoo, 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 pachoo. So plasma B cells can actually shoot out the antibodies toward whatever the invader is to combat the current infection. Memory B cells are like, I got you. And they remember the antigen. And their job is to just hang out and be like, I remember you. So that if that antigen ever comes back into the body again, the memory B cell is like, yo, I got this. I've seen that one. I know what we need. And the memory B cell is right there for you. Okay. So the naive B cell bonds. And then as soon as it bonds to that antigen, as soon as it realizes he's the right thing, she's the right thing, it will differentiate out into plasma B cells and memory B cells. I don't know why I was really worried about getting the right gender on those B cells, but at any rate, no matter what their gender is, or if they're, no gender, that's fine. Okay, so again, here we have B cells, and the B cells are different. Nope, not gonna let me write on it. Okay, thanks, dead stylus. So the B cells are all different. When the antigen binds, the antigen molecules can come in and bind. So in this case, the little red molecules are only able to bind with that middle B cell. Every naive B cell is different, but that middle one worked. And as soon as the antigen molecules bound, then bam, differentiation. So differentiation off into memory cells and then off into plasma B cells. And then those plasma B cells are able to shoot the antibodies out, pew, 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 toward the antigens in the body. All right. Now, remember, I also said there were T cells. So T cells have a job too. So you already heard about one kind of T cell, the helper T cells. The helper T cells are the ones who are like, oh my God, we need help. Okay. So the helper T cells recruited the B cells. But there are also T cells called cytotoxic T cells. So cyto means cell, toxic, I think you know what that means poisoning, and then T cells. So they're cytotoxic T cells. So they're T cells that go find infected body cells and kill the infected body cells. So in this image down here, again, and it's an electron micrograph, but it's colorized. So cancer cells aren't really that bright pink. But you have the cytotoxic T cell that's that yellowish orangish color that's climbing over and going to it. And what it's going to do is it's going to inject chemicals in that cancer cell and cause that cancer cell to die. Okay. So our cytotoxic T cell, once it recognizes the cancer cell, is able to go in, attack it, and kill it. Okay. So cytotoxic T cell binds to the infected cell. Okay. You can see that happening here. It's going to shoot these chemicals in to these holes. So perforin is going to make holes in the infected cell's membrane. An enzyme is going to go in there that's going to promote apoptosis. You know what apoptosis is now. It doesn't, and it's going to promote it. So apoptosis that wasn't going to happen now, we artificially induce that apoptosis, and then the infected cell ends up destroyed. So a cytotoxic T cell is going to go make a hole, put in the chemicals, and cause the cell to be destroyed. Okay, and so this is for infected T cells. So here's a video of a cytotoxic T cell.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells, and they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
Wasn't that awesome? All right. So memory T cells, just like memory B cells, they're there to remember, okay? But what they do is they remember what an infected cell looks like. So the next time they can kick into gear faster. So remember, a memory B cell remembered what the antigen looked like. A memory T cell remembers what an infected cell looks like, okay? The creation of T cells is considered a cell-mediated response. And it's called a cell-mediated response because it involves actual infected cells. So it's a response mediated or controlled by the infected cells. So T cells are cell-mediated, okay? The secretion of the B cells and the antibodies is considered a humoral response because the antibodies are distributed via the blood and the lymph, which we call the humors. So way back before we really understood as much as we understand now about the body, people talked about the humors. And so bile was one of your humors. Phlegm was a humor. Blood and lymph were humors, okay? So it's a humoral response because it's in the humors. So B cells, humoral response, T cells, cell-mediated response. So again, another way to have those branches in your graphic organizer. Okay, so B cells versus T cells. This is probably the biggest thing when you start talking about all the different names of things. So B cells, which are the humoral response, attack invaders that are outside of cells. They attack the actual invaders. T cells, I don't like the way this is looked. It's the cell-mediated response. And it says I attack invaders inside the cells. I don't really like that. I would like it better if it said I attack infected cells, okay? Because the cell itself is going to get attacked as well. So it's not like they go in, take out the invader, and leave the cell alone, and the cell is fine. The cell, again, another example, the cell is collateral damage. And so the cell that's been infected is going to go along with the invader. So it's not just the invader, okay? So again, B cells attack invaders, T cells attack infected cells. B cells, humoral response, T cells, cell-mediated response, okay? Now, that's all the first infection. So take a minute, pause the video, see if you can see how far you can get in running yourself through what happened with that first infection. Okay, how'd you do? Hopefully pretty well, okay? All right, now, second infection. So now that invader, so cold virus number 1 comes back into your body. So cold virus number 1 has already been in your body. You've taken care of it. Your body has figured it out, handled it. You've healed. You're better. Next winter comes along. Cold virus number 1 enters your body again. So this is our second infection. So now what's going to happen is those memory B cells are going to be like, yo, I've seen you before. They're going to recognize the antigen. They're going to get the signal. They're going to signal the plasma B cells, get them to posse up. They're going to start producing antibodies right away. We don't have to mess around with the naive B cells anymore. Okay, we got to figure it out. We don't need those guys. We got to figure it out. So the memory B cells hop in. They get the plasma B cells. Everybody starts attacking right away. The memory T cells do the same thing. They recognize the infected cells. They signal the cytotoxic T cells. They get to work. So all the infected cells get taken care of before they can perpetuate this illness further and further and further and further, okay? So in this graph, you can see, if you look so it looks like day 1 is where you've got the first exposure to your antigen, to antigen X, okay? And then we move over to day 7, so you've got that incubation period. You've got like a 6-day incubation period there. And so you make it to day 7, and at day 7, you start to get an immune response where your body's like, holy cow, there's somebody in here who doesn't belong, okay? So you've got this immune response now. And then your body is going to start to figure it out and start to produce antibodies. So it's going to produce all kinds of antibodies to antigen X. Then as you get healthier, you approach day 28, the antibody production goes off because you're healthy. You don't need it anymore, okay? If we expose you to antigen X again now, though, so now time has gone by, we expose you to antigen X at this point, your body is going to be like, whoa, we've seen this before. Pachoo! And so your antibody response is going to be faster and much larger. And so you're going to produce a ton of antibodies really fast, okay? And then that's going to taper off again, okay? So that first exposure takes a while. Second exposure, super quick. So lots of times you have viruses coming into your body that you've had before and you never even know it because your body is so fast at getting rid of them the second time, the third time, the fourth time, the fifth time. Now, if we bring in a different antigen, which is where that red line is, if we bring in a different antigen, your body, day 35, we bring in a different antigen, new antigen Y comes in, okay, 28. 28, we have first exposure, 28 days, I apologize, first exposure to antigen Y. Day 35, your body finally figures it out, and now we've got that first response again. So every antigen that comes in has to go through that process of a macrophage comes in, a macrophage gobbles it up, becomes an antigen for the hunting cell, goes to the helper T cells, the helper T cells go, oh my God, we need help. Naive B cells go, let me try, bingo. Naive B cells become plasma B cells and memory B cells. They let the cytotoxic, the helper T cells have also alerted the other T cells. And so the other T cells are going to let the helper T cells form memory. We're going to get memory T cells. And we're going to get cytotoxic T cells that are spurred on so that then they can go gobble those guys up. It's going to happen a lot faster the second time because the second time you don't have to do the whole thing with the naive B cells. Okay. So let's talk specifically about some other interesting things. Okay. So let's talk about some other things, bigger ideas with immunity. Okay. Active immunity means that we have memory cells that are present in the body after you fought off a pathogen, just because you got sick of it, just because you got sick with it. So you encountered that pathogen in your daily life and you fought it off. You could also get active immunity through a vaccine. So with a vaccine, what we do is we weaken whatever that pathogen is. So let's say it's the chickenpox virus. We're going to weaken the chickenpox virus and we're going to put the chickenpox virus into your body when you're little. Okay. When we do that, your body is like, oh my God, we're sick. And then your body figures out how to fight off the chickenpox virus. Truthfully, it was a really super wussy version of the chickenpox virus that we gave your body. And your body thinks it's like being awesome. And it's like, I'm so strong. Well, that's great. So we fooled your body. We told your body you were sick. You weren't really sick, but now your body knows how to get better if that chickenpox comes in. So we've created that first sickness so that now with the second sickness, you don't even know. Okay. So a vaccine is just fooling your body into thinking you're sick when really you're not. So you might get a little bit of an immune response after you have a vaccine. So some people will say, I had the flu vaccine and it gave me the flu. It did not give you the flu. If you had the flu, you would have been knocked on your butt. You just felt a little crappy for a day or two. And that's okay because that was your body showing you that it was fighting off whatever that thing was that was in the vaccine. There's also something called passive immunity. So passive immunity is when the antibodies can actually be used themselves to produce temporary immunity. So instead of your body having to go through the whole naive B cell thing and figure out what antibodies work, we can just give you the antibodies. So one way that happens is a pregnant woman gives the antibodies to her fetus through the placenta, or a mother can give the antibodies to her baby during breastfeeding. And so one of the reasons that people say you should really breastfeed for the first 6 months that a baby is alive is because that baby has a very weak immune system. And so if the mother can pass the antibodies on to the baby, then the baby has those antibodies that maybe it couldn't produce on its own. The problem is that those antibodies aren't going to stay in the system forever. They're just proteins. They're going to get broken down. They're going to get used up. They're going to get worn out. So they don't stay forever. Another thing you can do is inject antibodies. And so if you have some sort of infection that they're not going to let you ride it out until your body figures it out, they can actually inject the antibodies into your body so that your body can fight it off while your body figures out how to fight that off on its own. Again, the problem with passive immunity is it's not permanent. Active immunity is, because your body then remembers what to do and how to solve that problem for itself. So passive is great for the short term. It's not great for the long term. All right, so antibiotics. We know that antibiotics work on bacteria. They do not work on viruses, okay? Some types of antibiotics, so there are different ways that antibiotics work. Some types of antibiotics prevent bacteria from forming their cell walls. So if you've ever been on penicillin or amoxicillin or anything like that, they're going to keep bacteria from forming cell walls. And of course, if they don't have cell walls, they don't have a boundary around them, they're not really going to be able to replicate. And so your antibiotic is going to work. It's going to stop that reproduction. Some of them actually keep bacterial ribosomes from working. Okay, bacterial ribosomes, what does a ribosome do? Right, a ribosome makes proteins. And so if you keep the bacteria from making proteins, then you pretty much keep the bacteria from making the tool it needs to do almost everything to carry out its functions. So if you've ever been on erythromycin is an antibiotic that affects the bacterial ribosomes. Another kind actually breaks apart the bacterial DNA as it's forming. Okay, and it's going to prevent that DNA from being repaired. So Cipro is an example of an antibiotic that's going to keep the bacterial DNA from forming. Antibiotics, remember, only work on bacteria, not viruses. Remember that they work on good bacteria that we need, as well as the bacteria that are pathogens. So if we kill off all of those good bacteria, then if somebody's resistant, then we can allow those other guys to reproduce. So you could have long-term health issues because your gut needs those good bacteria, okay? You can also have bacteria that evolve into antibiotic-resistant strains that we can't kill. And we've talked about that before. All right, organ transplants. So sometimes organs get rejected because those transplanted cells have surface proteins that trigger an immune response. So that's why you have to have these tests if you want to be an organ donor, to make sure that your surface proteins and their surface proteins aren't going to conflict with each other. Because if we take your organ and put it into someone who doesn't have similar surface proteins, their antibodies are going to attack your organ. And that organ's going to end up having been completely useless because their body is going to think an invader is there and it's just going to go in and attack it and kill it. Autoimmune diseases, lupus, rheumatoid arthritis, multiple sclerosis. Those are examples of places where your immune system actually turns on your body's own cells. So the different reasons, if you want to read about each of those specifically, you can. There are different reasons for each, and I think we're still learning a ton about each of these. We don't know nearly enough about any of these, but basically your immune system is attacking your own body. So your immune system is turning on its own cells. And then AIDS. AIDS attacks the helper T cells. So those cells that were like, oh my God, there's an invader. AIDS, actually, the HIV virus actually attacks those cells. And so that's going to impair the humoral response, the B cells that get signaled, and the cell-mediated response, the cytotoxic T cells and the memory T cells. So if you go to the helper T cells, that's the juncture. And now your immune system can't work. And so most of the time, no, all of the time when people die, they don't die of AIDS. They die of pneumonia, or they die of a cold, or they die of some other infection that their body was not able to fight off. It's that the AIDS made it so that they could not fight off whatever that disease was. All right, so allergies. So you may have noticed earlier in the video that I said histamine. And some of you guys might have been like, wait a minute, but I could take antihistamines. Yes, this is going to key in here. So what an allergy is your body being extra, okay? An allergy is your body seeing something and going, oh my God, I can't believe you've come in here. This is terrible. This is bad. We have to attack it. Dog dander, cat dander, we simply cannot handle it. And your body freaks out. And I'm not judging. I'm allergic to like 9 billion things, okay? So allergies are just your body going, oh my God, I can't handle it, to something that really your body should be able to handle. It's your body being super melodramatic over something. So the antigens that cause allergies are called allergens. So take antigen and allergy, mush them together. Well, allergy and antigen. Allergen, okay? Gen means you're creating something. So they're allergens. They create an allergy. They give you this allergic reaction, okay? And they can actually occur in lots of different parts of your body. They can occur in your nasal passages, so you might get really stuffed up or have a runny nose. They can be in your bronchi, so it's really hard for you to breathe. They can be on your skin, so you get a rash or what looks like an infection of some sort. You've got this two-stage reaction. So one is that you're exposed to the allergen, and two, you get exposed to the allergen a second time. And when you get exposed to the allergen a second time, your mast cells reduce histamine. That histamine then is out there and causing this crazy response for you. So if you go back to that inflammatory response that the histamine triggered, you're basically getting the same thing. So if you want to take a medicine to make the histamine not do its job, what kind of medicine are you going to take? You're going to take an antihistamine. So an antihistamine is going to work against that histamine. So if you have allergies and you take an antihistamine, you are working against your body's own natural defenses, but it's okay because your body is throwing out its defenses towards something it doesn't need to throw its defenses out toward. If you are genuinely sick, you might not want to take an antihistamine because then you're fighting your body's natural defenses, and your body's natural defenses are trying to take care of this invader. And so you probably don't want to get in the way of your body's natural defenses with that. Okay. So here's a little graphic. There's fancy words here, but you've got your allergen pollen in this example, goes into the bloodstream. Your body freaks out and it goes, oh my God, and it makes antibodies for that. The antibodies attach to a mast cell. The allergens bind to the antibodies on the mast cell. When you get that new allergen that comes in, you release more histamine, and now you get these allergen symptoms. So you have the exposure, and then you have the later exposure that's gonna trigger that response. All right, so quick word about hand washing and hand sanitizer. Again, we've talked about this before when we talked about bacteria. Be sensible. Wash your hands after you use the bathroom, when you're going to handle food, after you have contact with somebody who's sick or if you're sick. Those are the times you need to hand wash. Use hand sanitizer if you don't have the ability to wash hands. But if you overdo it, you could end up with your hands that are really, really dry. They could be cracked open. You would actually end up opening up wounds that are going to allow pathogens in. So that skin barrier, that very first external barrier that was keeping pathogens out, now you've created a hole in it and you're allowing pathogens in. Okay. Here's a thing that's interesting is that when kids aren't exposed to antigens, their immune systems get kind of bored because there's nothing to do. So their immune systems just aren't active enough and they get kind of bored. And so what research is starting to find out is that kids who aren't exposed to enough antigens when they're little actually have higher rates of asthma and higher rates of allergies. I know that some of you guys, I'm not gonna say that. And then, of course, when we kill off the normal pathogens, we reduce the competition for the mutated pathogens. We increase the frequency of the resistant ones. We know that conjugation can happen, and we know that then we can end up with all of these other really terrible pathogens that we don't want to deal with. So there you go, your immune system in a really long lecture. Sorry, it is what it is. Go back and watch whatever parts you need to rewatch. Check your book. Look for other videos. I'm sure Khan Academy has great stuff on this. Go read what you need to read, and let me know if you have any questions.